There are many different types of drugs that produce sedation, even drugs whose primary use is for other purposes, like antihistamines. Each class of drug with sedative activity has a slightly different constellation of effects in addition to sedation. In this lecture, we're going to cover the effects of antipsychotic drugs related to acepromazine. When should you use them or shouldn't you use them? These drugs are characterized by a relatively long-term sedation with very little muscle relaxant and respiratory depressant effects. Their actions are brought about by their ability to inhibit monoaminergic activity by a competitive blockade of their receptors. Monoamines are compounds like dopamine, norepinephrine, and serotonin. You can see that their structures are very similar to each other, so their receptors are also very similar in their binding regions. Norepinephrine is the same compound as noradrenaline, just as epinephrine is the same as adrenaline. There are two major types of antipsychotic drugs, also known as major tranquilizers, that are used widely in clinical situations. One group has a phenothiazine-based chemical structure, and the other group is based on a butyrylphenone structure. In veterinary medicine, acepromazine, also known as ACE, is the most widely used phenothiazine, and droperidol is the most widely used butyrylphenone. Phenothiazine itself, with no substitutions, was originally used as an insecticide and anthelmintic compound. It was in this capacity that it was found to produce sedation. Substitutions on these molecules optimize either their sedative or antipsychotic activity. So most older, traditional tranquilizers are derivatives of these two structures. As you can see from their structures, both types of molecule are fairly large, rigid compounds. The rule of thumb in pharmacology is that large, bulky, rigid molecules tend to be antagonists, whereas smaller, more flexible molecules that bind to the same receptor tend to be agonists. Based on this, you can predict that major tranquilizers are competitive antagonists at monoamine receptors, whereas the smaller, more flexible structures like monoamines themselves, here it's norepinephrine, are agonists. Depending on the chemical substitutions added to the phenothiazine or butyrylphenone molecule, a variety of compounds can be synthesized, each with its own unique set of actions and uses. Because of this, different drugs are often preferred in different species. Some of them are available as a single drug preparation, and some come in combination with either a glucocorticoid, in the case of trimeprazine when it's combined with prednisolone, or together with an opioid compound, as in the combination of either imobilon, a combination of ACE and etorphine, or inovarvet, a combination of droperidol and fentanyl. So what are the pathways in the brain that are affected by these drugs? Dopamine is a major tranquilizer in the brain. It's synthesized and released from dopaminergic neurons that project to the cortex, the midbrain, and the spinal cord. Importantly, dopamine is released in the accumbens and amygdala, where it regulates perception and behavior associated with psychoses. It's also released in the striatum, where it regulates motor activity, and in the chemoreceptor trigger zone, where it stimulates vomiting, that is, emesis. Too much dopaminergic activity can lead to psychotic activity, abnormal control of movement, and emesis. By inhibiting dopaminergic activity in the brain, acepromazine has antipsychotic activity and provides antiemetic activity, but it also induces catalepsy, which is an abnormal stance or posture. In addition to its blockade of dopaminergic receptors, drugs like acepromazine inhibit noradrenergic activity in the reticular activating system. It's by this action that sedation is produced. Drugs that have the greatest ability to inhibit norepinephrine are the most sedative and the most widely used in veterinary medicine. In addition to antagonizing norepinephrine, the anticholinergic and antihistaminic effects of some drugs also contribute to their sedation. What is the major clinical use of these drugs in humans? They're mainly used for their antipsychotic activity and to a lesser extent for their sedative effects. People with psychoses have excessive dopaminergic activity in areas like the accumbens and amygdala, so drugs used in humans are selected for their potent antagonism of dopamine. For these purposes, anti-adrenergic activity, which produces sedation, is considered an undesirable side effect. So the clinical significance is that drugs selected for use in humans are generally different than those selected for use in veterinary medicine. While some animals may be psychotic, we can't diagnose a dissociation from reality in organisms that can't talk. So when used in veterinary medicine for behavior modification, it's on a trial and error basis, and it's not clear whether it's the antipsychotic effect, blocking dopamine, or the sedative effect, blocking norepinephrine, that's effective. So what are the major clinical uses of major tranquilizers in veterinary medicine? Most importantly, they provide sedation prior to anesthesia. The most commonly used drug for this purpose is acepromazine, otherwise known as ACE. During anesthesia, they promote a smooth induction and recovery in situations where the anesthetic alone typically produces a rough induction and recovery. They also reduce the dose of anesthetics and narcotic analgesics required during surgical procedures. Because of this, they reduce the occurrence of side effects. Some of these drugs have anticholinergic properties. This reduces and even eliminates the need for atropine-like compounds in species that typically receive them as part of their pre-anesthetic protocol. This atropine-like effect not only prevents excessive salivation and so forth, but also protects against cardiac arrhythmias induced by many of the halogenated anesthetic compounds. Acepromazine type drugs also protect against vomiting, that is emesis, in response to anesthetics, like the effect of metoclopramide, which is newer and a more specific D2 receptor antagonist. They can also be used alone to treat motion sickness, like maropitant, a neurokinin 1 antagonist. Another whole category of use is chemical restraint of exotic species in zoo medicine, where they're typically administered together with narcotic analgesics, that is, opioids. Some are antihistaminic, and because of this, antipyretic, that is, they inhibit itch, especially when delivered in combination with glucocorticoids like prednisolone that have anti-inflammatory actions. Finally, they're used for behavior modification in a variety of situations, such as excessive barking or aggression. Now that you know some of the situations in which these drugs might be used, how are they used, and what might you expect when they're injected? They're absorbed either orally or parenterally, but they're usually delivered IV or IM to produce as rapid an effect as possible. This is because they take about 10 to 15 minutes to reach their peak sedative effect, even after an intravenous injection. Compared to other sedatives, major tranquilizers generally have a slow onset of action. In some situations, you can tell when the drug is sufficiently effective to begin a procedure by the side effects produced. For example, in horses, relaxation of the retractor penis muscle is a good marker in stallions. In dogs, relaxation of the nictitating membrane of the eye is a good indicator of a systemic effect. They're metabolized slowly in the liver, giving them a very long half-life and subsequently a long duration of action, longer than most other sedatives. Metabolites are uneventfully excreted in the urine and bile, so excretion has no effect on duration of action. These drugs are distributed widely throughout the body. They have a high lipid solubility, resulting in about 90% bound to plasma proteins. This binding decreases their availability as only the free, unbound drug is available to enter the brain. If used chronically, they may also tend to accumulate in the brain and lung by binding to their proteins, a fact that may influence their long duration of action. Finally, they're able to cross into the placenta and reach the fetus, but unlike some drugs, they can be used during delivery because they produce less respiratory depression of the offspring than other sedative compounds. Given these characteristics, the next video will consider some of the implications on the clinical use of these compounds.